All right, welcome to the second lecture from Chapter 18. So in Chapter 18, in the first part, we discussed the electron transport chain, and in the second part, we will discuss the process of oxidative phosphorylation. So just as a review, when we talked about the electron transport chain, we discussed how all of our reduced electron carriers from glucose catabolism could be used to help generate a proton gradient. During oxidative phosphorylation, we'll show how ATP synthase uses that proton gradient, to help make ATP. So first we'll take a look at the structure of the ATP synthase enzyme. Then we'll talk about its mechanism. And finally, we'll discuss all of the energetics overall that are used for the generation of ATP. So first, as just a general overview of ATP synthase, it is located in the inner mitochondrial membrane, just like the components of the electron transport chain. It uses the proton gradient that was generated by the electron transport chain to help drive the synthesis of ATP, which can be used as an energy currency by the cell. And as you learned in Chapter 10, ATP synthase is an F-type ATPase. The two links here on the slides are to some animations that have explanations and show you using a video format how ATP is generated by ATP synthase. Since there are a lot of moving parts in ATP synthase, it might be helpful for you to watch these videos to see how they're generated in a slightly different way than the way I will show you in these slides, which is just through pictures. So if you find it helpful to watch videos, these are recommended. So with that said, let's take a look at the structure of ATP synthase. So this on the right is a picture of ATP synthase from the side. It's the E. coli ATP synthase. Overall, a lot of organisms require the generation of ATP with oxidative phosphorylation, and so ATP synthase is really similar across a wide variety of organisms. So even though this one is from E. coli, it's not that different from the one that's inside our cells as humans. So there are two main domains of ATP synthase. First is the F0 domain. This domain is the proton pump, and it is embedded in the cell membrane. So this is the transmembrane domain of ATP synthase. And then the F1 domain, shown here at the top, extends into the matrix of the mitochondria. So like we mentioned, this protein ATP synthase is embedded in the inner mitochondrial membrane. So the F0 domain is in the inner mitochondrial membrane, and the F1 domain extends out into the matrix. And as we mentioned, the proton pumping that was done during the electron transport chain pumped all those protons into the intermembrane space. So in the intermembrane space, at this point when ATP synthase is ready to be used, there are a bunch of protons that have been pumped into there out from the matrix. And so the ATP synthase is going to use this passing of the protons back down the gradient back into the matrix to help drive the synthesis of ATP. And the way it does that is with the combination of all these different subunits. So in the F0 domain, the transmembrane domain, there are A, B, and C subunits. In particular, there are a whole bunch of C subunits that help to take up those protons from the intermembrane space and then pass them. Those protons are going to get passed down the epsilon and gamma subunits that are in the F1 domain into the actual matrix portion, and then into the part of the ATP synthase that actually helps to directly catalyze ATP synthase, which is actually these beta subunits. There are also alpha subunits that are really similar structurally to the beta subunits, and they are located all together, and we'll see how that looks a little bit on the next slide. But it's the beta subunits that are actually directly catalyzing the synthesis of ATP. So overall, in the F0 region, there are A, B, and C subunits. And in the F1 domain, there are alpha, beta, gamma, delta, and epsilon subunits. All right, so this is our side view. So keep that in mind as we flip it over and take that top view. So from this top view, it's a lot easier to see the arrangement of the alpha and the beta subunits, which are alternating. So there are 3 alphas and 3 betas, which I'll show in a different color. And if you look down in the middle here, we see this dark blue region. That is actually, if we look back, this is the C the subunits that are part of the F0 domain that are located in the membrane, because we're taking a top-down view here. And this helps to give you a sense of this whole thing is sort of going to be rotating, and this rotation is going to help drive ATP synthesis, which we'll see in a little bit. And so let's see how this rotation helps to drive conformational changes within the ATP synthase subunits that ultimately helps to make ATP. So first of all, let's think about which subunits are remaining stationary. So from the F0 domain, A and B are remaining stationary. It's C that's going to rotate. From the F1 domain, it's delta and the alphas and betas that remain stationary, but it's gamma and epsilon that are going to rotate. And so see, gamma and epsilon are here in this sort of rod, and C is here in this sort of ring. So that structure is going to rotate, while alpha and beta up here are going to remain still, as well as everything else. So let's look at what is actually happening to generate ATP now. So as we recall, there are a whole bunch of protons that have been pumped into the intermembrane space. So one proton at a time from the intermembrane space is going to pass into the C ring through the A subunit. There's a space here. And one proton can enter each one C subunit. And because of the charge distribution that's already in each C subunit, when a proton is added, that's going to make it a little bit more hydrophobic, a little bit more nonpolar. And so that piece of the C subunit is then able to start to rotate into the hydrophobic portion of the intermitochondrial membrane, because adding that proton helps make that C subunit want to react more with those nonpolar regions. So ultimately, as protons are getting passed into these C subunits, they're driving some small amount of rotation. And as the C subunits are rotating, so are the gamma and epsilon regions that are attached to it. And eventually, when that C ring that's made of all these different C subunits goes all the way almost fully around a full turn, finally that proton is able to escape out into the matrix, which is the proton passing down its gradient. In that process, like I mentioned, the rotation in particular of this gamma subunit is going to switch the beta subunits in between 3 different conformations. And these conformations are called tight, loose, and open. And these tight, loose, and open conformations are actually what is going to synthesize and release ATP. And we'll look a little bit more about how that works on the next slide. So, like we mentioned, there are 3 different states, open, loose, and tight. And so we start out with ADP and an inorganic phosphate group are going to bind in the L binding site, a loose binding site of one of those beta subunits. And for this reason, the loose binding site is also called beta DP, D for ADP and P for phosphate. And so when that ADP and inorganic phosphate enter into the loose subunit, at that point, or meanwhile, right, this passage of protons through the C ring is going to be causing this change in conformation of this rotation of this central portion, which is our gamma portion, which is going to help drive this conformation of our subunit that we're looking at on top. So we have this change now from L, which is loose, into T, which is tight. And so when we have that change from L to T, at this point, this sort of tight conformation helps to push together the ADP and the inorganic phosphate, forming an ATP out of that ADP plus inorganic phosphate. And then as that subunit keeps turning, the T site then converts into an open site, which is also called beta E. And this will finally allow the ATP to be released. And so over and over, as the C subunit is turning, it's turning this that little rod, the gamma and epsilon, which is helping to drive this change in conformation of these 3 beta subunits. And again, these beta subunits are what's letting ADP plus phosphate being turned into a molecule of ATP. So overall, let's look at those energetics. So for like we mentioned, for every 1 full turn of the C ring, it's going to accept a number of protons, and that number of protons depends on how many C subunits there are in that C ring. And this depends on the organism and their particular ATP synthase. For us, it's about 10 protons. So there are about 10 C subunits in an ATP synthase for every ATP synthase molecule. About 10 protons can enter to cause the ATP synthase to rotate one full turn. And each full turn is going to generate 3 ATPs because one ATP is generated for each beta subunit, and there are 3 beta subunits in each ATP synthase. So one full turn requires 10 protons to move down their gradient and is going to generate 3 ATP molecules. And with this information now, we can calculate the theoretical number of ATPs that can be formed from one NADH and from one FADH2, assuming that there are 10 C subunits in one ATP synthase. So let's take a look at how that works. So for NADH, we talked about in the last lecture how 10 protons are going to be generated from each NADH molecule through the electron transport chain. We also have our conversion factor for ATP synthase, where 10 protons are required to generate 3 ATPs. And so if you multiply this out, the protons are going to cancel with each other. So ultimately, one NADH reduced electron carrier is able to generate 3 molecules of ATP, theoretically. For FADH2, we talked about how this is going to generate only 6 protons in our proton gradient because of the way it enters the electron transport chain. And then if we multiply again by our conversion factor, which is 3 ATPs for every 10 protons, we can calculate that for every 1 FADH2 reduced electron carrier, we can generate, theoretically, 1.8 ATPs. But this is the theoretical sort of maximum number that can be generated in a perfect world. But the maximum number of ATP is not usually synthesized for a number of reasons. So one reason is that protons are able to just slowly leak back through the membrane down their gradient, so from the intermembrane space to the matrix, without passing through ATP synthase, so they are not helping to drive that rotational force. Also that proton gradient can help to drive other machinery in the mitochondria to help, for example, to drive transport. So not all the protons that are used to create this gradient from NADH and FADH2 are then actually put back into driving ATP synthase. And so that's why the maximum number of ATP doesn't ever actually get synthesized. So let's look at the actual numbers for NADH and FADH2. So for NADH, we talked about theoretically you can get up to 3. In real life, generally one NADH will give you about 2.5 ATPs. For FADH2, we just talked about the theoretical number is 1.8 ATP at the maximum, but in reality, it's really more like 1.5 ATPs for every FADH2. And it's important to know this and sort of understand where this comes from and how this all works and how everything kind of all ties in together from all of our different steps of catabolism that we discussed in the last few chapters. So overall, now you are able to answer the practice questions that are about oxidative phosphorylation, and you have also completed the last video for this class. So congratulations.